Our first speaker has lived in 4 countries before the 6th grade. So I do think we're going to have to ask what those 4 countries were. Everyone, please welcome Simina.
Hello, I'm really happy to be here. And thank you so much for inviting me and giving me the opportunity to talk to you about some of my experience in using R to study rare diseases. And I will be talking about Duchenne muscular dystrophy specifically, but I hope that this will be an opportunity for everybody to become familiar with some of the ideas and also some of the ways in which you can ask and answer questions in rare diseases. If you're interested in getting in contact with me, the easiest thing to do is probably Twitter. And actually, if you go, my pinned tweet on Twitter is actually linked to this presentation on GitHub. So you can have all this information, including my email, if you just go ahead and do that. Okay, so just a quick introduction to rare diseases, which generally have this this zebra ribbon, in case you've seen it. Sometimes it refers to specific diseases, sometimes to just rare diseases in general. So a rare disease in the United States, it's often defined as a disease that affects fewer than 200,000 people, which is a fair number of people, but of course it's very small compared to the US population. And over 70% of them are genetic. Unfortunately, that means that a lot of them do affect children, especially rare diseases which are recessive. So basically you need a copy of a gene that has some issues from both parents. Those tend to affect children more often. And they lead to a substantially reduced life expectancy. I think that they used to be called fatal, but now I think the correct term that people generally use is life-limiting. And some examples that people might be familiar with include cystic fibrosis, sickle cell anemia. Some of them, if they're dominant, like Huntington's, they might actually affect adults, but oftentimes they affect children, and muscular dystrophies, and I'll be talking about one of them, Duchenne muscular dystrophy, or DMD. Now, one thing about rare diseases that rare disease advocates really like to point out is that even though individually they're rare, as a whole, they actually affect more than 10% of the population. I guess close to 10% of the population. It might actually be more than that because some of them, of course, might be undiagnosed or underdiagnosed due to their rarity. So the question is, what are some approaches for studying rare diseases? And of course, since it's the R conference, how can we use R? And what are, kind of some of the priorities in doing this? So just a quick little tidbit about this ribbon. Why is it the zebra ribbon? So Theodore Woodward, who was a very well-known and accomplished researcher at the University of Maryland, so he said, when you hear hoofbeats behind you, don't expect to see a zebra. And I guess this is especially because in Maryland, where I live, for example, in Maryland, there are not that many zebras, right? Of course, it depends like where you are, right? Context is everything. The idea is that, for example, if you're a doctor, if you see a patient, even if they kind of seem to have rare symptoms, they're more likely to have a common disease, just because of the numbers, right? And so the idea is that, rare disease advocates do want to point out that, there are zebras out there. So they sometimes think of themselves as, medical zebras. And the other thing is that each zebra is also unique in terms of their striped pattern. So just something that I just wanted to share. So some challenges with rare disease. So there's very many challenges, including, of course, it can be very hard to diagnose them, right? Because there is this issue of, kind of being like a, quote unquote, a zebra. And it depends on what the disease is. I mean, there's screening now, newborn screening for some of them, but of course that's a challenge. But even post-diagnosis, of course, it's an issue in terms of just performing studies and performing clinical trials, for example. So the obvious reason, of course, is that, of course, there's a very small number of individuals affected by any one disease. And so your sample size by definition is going to be low. So that's going to, of course, be a problem from a statistical point of view. They also oftentimes have a progressive nature, unfortunately. I mean, it does depend on the disease, but it is often progressive. And so there's this problem of what clinical outcomes should you consider, because unfortunately, as the disease progresses, so for example, for Duchenne muscular dystrophy, it's a muscle wasting disease. So when children are young, typically the outcomes are how much can they walk in 6 minutes. That's one of the outcomes that's considered a 6-minute walk test. But as it progresses and you start having the heart being affected and the respiratory system being affected, and people can no longer walk, then you can't really use that outcome anymore, right? So there's this issue of like, how can you use outcomes? And then depending on how old people are, there's different outcomes that are relevant and your sample size can be reduced even more. And of course, if you're performing research on children, there's ethical considerations. And also in terms of implementational challenges, I think those of us who either have children or work with children know that it's very hard to tell like a 5-year-old, for example, please walk for 6 minutes, but don't run, make sure you walk for 6 minutes, right? And because of some of these reasons, the FDA does actually have some specific flexibilities when they evaluate new therapies for rare diseases, for example, by allowing surrogate biomarkers to be used instead of specific endpoints. So maybe looking at the impact of a drug on a protein, or I'll be talking about the impact of a drug on a metabolite, which is a small molecule. And if you can show that that's correlated with a disease endpoint, then you can just use that instead of the actual disease endpoint in the clinical trial. So, I mentioned I'll be talking about DMD or Duchenne muscular dystrophy. So it's an X-linked disorder. It's a very devastating disorder. X-linked means that it's caused by mutations in a gene on the X chromosome. So this is why it affects about 1 in 5,000 newborn males, a lot fewer females, just because most males have XY chromosomes. Of course, not everybody, but most of them do. And so if they have a mutation in a gene on the X chromosome, they can't have another X chromosome like females usually do to kind of compensate for that. So it can happen in females as well, depending on, if they have mutations in both, or if they have some part that's missing, or, sort of different situations. And there's actually work to see whether women who are carriers might have some of the symptoms, like maybe some cardiac symptoms as well. But in general, people think of it as being a disease that overwhelmingly affects boys. And even though it's caused by mutations in a single gene, it's actually the biggest gene in the human genome, as far as we know. And there's actually many different kinds of mutations that can happen in that single gene, and different mutations can actually lead to different phenotypes. So certain mutations can actually lead to a milder phenotype that's called Becker muscular dystrophy. They used to be considered 2 different diseases before people actually figured out the genetics. And there are therapies right now, some of which are in trial, some of which are actually conditionally approved, that only target individuals with specific mutations. So that's just sort of like, it's like a subclass of a small class of rare diseases. And it's an example of personalized medicine. And, that's all great. But again, it does complicate the situation, for example, of clinical trials. And then I mentioned, it leads to muscle loss, and generally, unfortunately, it leads to death. It's pretty rare to live, I think, past like early 30s. Some people live into their late 30s, but it's pretty rare. And the mainstay treatment is still steroid treatment, because that can help maintain muscle mass, but it can actually cause issues in the long term. And it's only good for, maybe like a few years. Okay, so I mentioned, so there are a few therapies that are approved or conditionally approved. There's many clinical trials right now actually for DMD, so that's great. But again, it can in some ways actually exacerbate this issue where you already have a small number of patients and you're actually having them be competed for even more in these multiple trials. And then typically I think families do actually try to enroll their children in these trials. But there is this issue where at a certain point, like they can kind of like age out. So, there's typically an age limit for various trials. And again, it depends on the clinical outcomes, on the clinical symptoms. It depends on the mutation type. So that is a challenge. So one thing that, I've started to get interested in is to actually look to see what trials are actually performed in this space, and eventually have the goal of developing a product that actually allows everybody who's interested in this area to stay up to date with the ongoing development, and also to try to prioritize research focusing on individuals who maybe are at greatest need. And so that you don't have like all the trials focusing on maybe the 13% of individuals who have a mutation that's amenable to exon 51 skipping, for example. And so I had a summer student, Bin Hai, last year who looked at clinicaltrials.gov, which is a repository of all clinical trials. Basically, every clinical trial, at least in the US, has to be pre-registered, and all of the files are XML files. And so she downloaded all the XML files that were related to Duchenne muscular dystrophy. And the great thing about R, of course, is that there are all these packages that allow you to interact with different kinds of files. So even though, usually I think about using like tab delimited or comma delimited files, even if they're XML files, you can still use the XML package, and you can actually pick out some of the blocks, the kind of the predefined blocks. And then of course, perform data cleaning. That includes things like redundant drug names. Every drug I think has at least like 4 names. That's a whole sort of issue onto itself. And then she used RShiny to design and create a web interface to explore these trials and actually look at, you can search by condition, and you can get some summary statistics and so on. And again, if you go to my Twitter account and looked at the pinned tweet, you can get to the slides, and you can if you want to play around with this and take a look at it. Okay, so the second vignette that I wanted to talk about was using molecular data sets to try to help out with actually finding biomarkers of either disease progression or drug response. So I mentioned that there's a focus in doing this, especially for rare diseases, and we focus on looking for metabolic biomarkers. So basically metabolites, which are small molecules. The study of metabolites is called metabolomics. They can be detected in various biofluids and tissues. That includes things like blood, urine, and so forth. And they have various functions. And the good thing about them is they can be influenced by both genetics and environment. So they're sort of like sit somewhere in between. So it's good to have this opportunity to look for different kinds of markers. And I'll be focusing on this first paper that we worked on from 2016. So this paper looked at 51 DMD patients and 22 healthy controls. We detected over 2,000 metabolites. The samples were all run at Georgetown in terms of the metabolomics, but they are from 5 different study sites. So one of the things that we were always we were careful to assess is to see whether there's any possible batch effects. And you can see actually the individuals are not balanced between the study sites. So we did also have also a sub-analysis just looking at Calgary, Alberta Children's Hospital, which did actually have a more balanced number of patients and healthy controls. So the first step, I always like to point out the first step in doing any sort of omics is first of all, doing a lot of data exploration, making sure that everything sort of matches up. And then I guess the second step is to do principal component analysis, because I think that it's very revealing in terms of, are there batch effects? Are there artifacts? When people see, for example, 2 clusters, sometimes they get very excited, like, well, I've discovered a pattern in my data. For me, it always just makes me feel nervous because I worry like, oh, does it mean that actually the samples are processed differently? So we were actually pretty happy to see this. This just color codes by age and this color codes by study site. Now, if I was to redo it now, I would redo it a little bit differently. I just wanted to kind of have this like as a parenthesis because I feel like I have learned something since 2016. So I do try to use a colorblind friendly palette because I'm not colorblind myself, so it's not something that comes automatically, but I think about 4% of the population is colorblind. So there's one of these questions like, how can we kind of better automate these things so you don't have to make these decisions, right? Which is kind of like tiring. And like, I always like kind of like going back and changing the colorblind friendly palette. I'm trying to get better at this. And then for PCA now, because if it's all omics, so unless you're using different types of measurements with different units, the recommendation is generally to actually not scale. So to use the default, for example, if you're doing print comp in R, and also to use the same distances on both axes. So you can see like here between 0, this is 0 and 10 on the y-axis, 0 and 10 on the x-axis, because the first principal component has more variability than the second principal component. So it's actually ideal to try to maintain that as opposed to having it be a square. And I learned a lot of these tips from this great Nguyen and Holmes paper, which is also a great example of reproducibility because they actually have the code for generating all of the figures in their paper. So I just wanted to just share that. Okay, so in our study, so basically we had over 2,000 metabolites. We basically fit a regression model for each of them that had as the outcome a transformed metabolite value, log transformed, various other quantile normalized, and so on. And then we looked at how that value can be explained by age, study site, and then disease status, of course, and then also age by disease interaction. And we found 14 metabolites that were significantly altered between DMD and healthy controls at a false discovery rate of 1%. Out of those 14, because of the technology that was used, we didn't know exactly what they were. We just had an idea of what their weight was and some other characteristics. So the lab that we worked with, which is the core at Georgetown, did validate them using MSMS, and they found 4 of them, creatine, creatinine, 5-ADHT, and testosterone sulfate. We're pretty sure that this is actually what they are, and L-arginine was likely validated. And this is actually, it doesn't seem like a great yield, but it's actually pretty common for these kinds of experiments. It's sort of like part of the course, like most things actually don't get validated. Creatine and creatinine, we were happy to see because we know that they're actually involved in the creatine pathway, which has to do with things that happen in muscle and in blood. And you see creatine and creatinine here. So these are both metabolites, they're small molecules. And this reaction here is catalyzed by creatine kinase. Creatine kinase is a protein, so we didn't pick it up in our study because it's too large. But actually our team did do, some members of our team did actually do a proteomics study where they picked up creatine kinase. And this is actually a very well-known biomarker a very well-known diagnostic biomarker of DMD. So it shows up being at very high levels in sort of children between the ages of maybe like 4 and 10 or so, because it shows that there's a lot of like muscle damage going on. So typically, if somebody is suspected of having DMD, they'll have a blood test where they look at CK levels, and typically those will be way higher than in healthy individuals. But the problem is that with age, as there's muscle wasting going on, and so with age that actually goes down a lot, and so you can't really use it as a biomarker of disease progression or of therapy response. But in any case, it was good to see these as some of our among our results. And we also considered in a post hoc fashion the ratio between creatine and creatinine, because you see that like one of them leads to the other, so they're actually inversely related. So their ratio is actually even more it's actually even more compelling to look at it. So you can see between DMD and controls, they're generally very different, and it has a very low p-value. But I mean, this was a kind of a post hoc analysis, but we were actually happy to see this result validated by another group. So that was good. Another thing I want to talk about, so in terms of doing this work, I think it's important to keep in mind that there is kind of this, I call it like a study ecosystem, like what's going on beyond the paper. And you can think of this as sort of being related to reproducibility, reproducibility powered by R. I think that's one of the big benefits of R. So I know some people have said that a scientific article or a report, it's sort of like advertising the research, because you're only kind of getting maybe like a big picture view, finding out, about like specific decisions that were made, maybe in order to have a certain result or a certain publication. But there's a lot that's actually kind of going on behind the scenes. This is published in PLOS, and so PLOS always wanted to deposit the data, as much as possible. And so we deposited the raw data in Dryad, which is one of the repositories that they recommend. And then the process data along with the analysis code is on GitHub. And they're all linked. Ideally, you can get all the links. So they're all linked, for example, from my website. I think it would be great to figure out some better ways as a community to sort of connect all these things. I mean, there's also like links, in the paper, but it's one of those things where, again, I think if you kind of incentivize it and sort of make it be automatic so that people don't have to kind of think about doing all of these things and kind of figure out, like every time, like I update my CV, it's like you have to like kind of copy and paste like 5 different links. I think it would be great to have a more automated way of doing that. Just like it would be great to have maybe like, like colorblind friendly palettes as just being the default, so you don't have to think through it and make more decisions about it yourself. So just some of the conclusions. So primarily for the metabolomic study, so the 2016 study that I talked about, we're very proud that it was the first comprehensive metabolomic study for Duchenne muscular dystrophy, and we thought it was one of the first steps in finding metabolic surrogate biomarkers and adding to this list, because people have looked at other kinds of like proteomics and looking at mRNA markers and so on. And we also had a 2020 study which looked at the urine of mice. So there's a mouse model called the MDX model, and we compared healthy mice with MDX mice, and then they either were just taking a control or they were on prednisolone, which is a type of corticosteroid. So we got some additional biomarkers from that as well. So some future goals in terms of also combining this with what I talked about in terms of clinical trials. I think it would be really great to have these types of studies embedded into clinical trials, because clinical trials have very clean data. They have very good clinical outcomes. And so then I think it's a lot easier if you can just have access to some of these samples where people have already, the people who set up the clinical trials are already measuring a lot of these outcomes on the study participants. And then you can also do kind of some omics sort of like on the side. I mean, it has to be embedded into the protocol, but I think that would be sort of like the ideal thing to do. And then also try to connect this type of study with genetic data and focus it on some of the, I mentioned the areas of greatest need in terms of maybe looking at people who aren't served by current trials in some way, right? Either because they have a rare mutation or because of their age or because of some other aspects. Okay, so I just wanted to acknowledge all the people who are involved in the study, our funding, the study participants, including these are 3 students who helped me out. They weren't involved in the original publications because they started working with me on this afterwards, but Bin was involved in the clinical trials work, and then Boyun and Shintong are looking more at the metabolomics data. We also did some metabolomics proteomics integration. Again, if anybody's interested, definitely feel free to contact me afterwards if anybody wants to talk more about it. And this is just my final advertisement slide. So we have a master's program in health informatics and data science at Georgetown. It's a great program, if I say so myself. I'm one of the faculty in it. We're in our second year right now, so we're open for applications for next year. Definitely let me know. It also includes, it's a one-year program that includes a summer sort of internship with industry, which is co-mentored by a faculty member as well. And, I recently joined Our Ladies DC as a co-organizer, and I know that Our Ladies is also involved in setting up this meeting. So these are some of the Twitter accounts for Our Ladies Global and Our Ladies DC. And of course, there are Our Ladies chapters in many cities in the US and the world. So thank you so much.
Thank you very much. That does remind me, thank you for the reminder, we are going to have an Our Ladies photo. Let me just check when that is supposed to be.